So I'm Tathagata Pal. I'm doing my final year PhD in the Indian Institute of Technology, Bombay. So my supervisor is Professor Somu Mukherjee. And the topic of my presentation is detection of arsenic in water by UV optical fiber localized surface plasmon resonance after sensor. I know it's a quite long title, so I'll break down everything in the next slides. And we are biosensor and bioinstrumentation laboratory. As the name indicates, we develop biosensor. So that will be the outline of my presentation. So first there will be an introduction of why arsenic detection is required. And there will be a schematic of the fiber optic sensing scheme. Then why did we choose aptamer and the requirement for the folding buffer? We have used two kinds of nanoparticles. One is cited cap gold nanoparticle, which is negative in charge, and system in cap gold nanoparticle, which is positive in charge. So I'll explain why we got finally results using the positively charged nanoparticles. Then I'll describe the sensing results in real matrices, tap water and lake water, and also the matrix vector, and also show the interference study, finally the conclusion and future work. So arsenic is one of the most toxic element in the periodic table. It is a group 1 carcinogen and it is a proven cancer causing agent. So this diagram shows all the acute and chronic effect of arsenic toxicity on human health. Arsenic exists in inorganic and organic forms. Inorganic forms is usually more toxic than the organic. Arsenite H3 is considered the most toxic among all other oxyanides. WHO has set a maximum permissible limit of 10 parts per billion for arsenic for human intake. Now, the arsenic contamination is mostly geogenic. It's not by human activity, so you cannot control it. So this graph shows the arsenic affected area throughout the world. So it is estimated by this group that around 230 millions of people are living in arsenic contaminated region, and that spans around 108 countries. And also the suspected region increases as the testing of the arsenic amplifies. The gold standard method for arsenic detection is ICP-MS or AES, which is quite expensive. It needs trained personnel. The sample preparation involves highly toxic chemicals. It's quite hazardous to the environment. And usually it is available in only high funded labs. So obviously it is not suitable for routine screening. So that brings to a need for field deployable, user-friendly, inexpensive sensor for wide-scale deployment. Okay. Till now we have seen in other presentations, usually electrochemical biosensor. Now here we are presenting, it's called fiber optic biosensor. So we all know that light travels to an optical fiber by total internal reflection. However, on the surface, there is a wave called evanescent wave. Evanescent wave is very sensitive to the change of RI to the environment. Now, when we bend the optical fiber into U shape, the depth of penetration of the evanescent wave further increases and gives more sensitivity to RI. Now, to enhance the sensitivity even further, we immobilize the optical fiber, this U shape, by gold nanoparticle. Gold nanoparticle gives LSPR effect, localized surface plasmon resonance. This further increases the sensitivity of the sensor. So this is the overall setup. There is a light source. It has a constant current, 120 milliampere current source. It is collimated through a collimator lens, and the light is basically white LED. It passes to the fiber. Here, when the analyte binds to the bounded receptor, we get a change of absorbance as a signal. And finally, the absorbance is measured through a spectrophotometer. This whole setup has been miniatured in our lab. We call it MuSense. So the device cost around 185 USD. And the sensor we can put like a cartridge. So this is the human optical fiber. The length is around 1.5 millimeter. And this is the holder. And finally, we put it in a cartridge, and cartridge goes into the device and we do the sensing. I have a video at the end of my presentation. So the fiber is basically purchased from Thor Lab. It is a multimode optical fiber. We remove the jacket, then we do the cleaning thoroughly, first by NOH to etching the fiber, then by HCl and methanol to remove all the organic debris, and finally we treat it with H2SO4 to express the silanol group. Finally, it is silanized by aminosilane and GNP is bound on the surface. Finally, again, aptamer is bound. I'll go into the details, and the rest of the site is blocked and it is ready for sensing. Okay, first, why do we choose aptamer for these sensing experiments? Arsenic is non-immunogenic, so we cannot have an antibody for arsenic. There are two enzymes. One is arsenite oxidase, another is arsenate reductase. Both are bacterial enzyme, and they are not available off the shelf. As we are talking about field evaluation sensor, we need in bulk. That's why we search for aptamer. We are not aptamer specialized lab, so we got this group when et al. They published a paper by electrochemical sensing. These aptamer sequence is specific to arsenite, and we purchased it from Sigma Electrics. It is thiolanded. So this is the form we received from the Sigma. First, we reduced the bond using TCEP. So that produced free thiol, and it also produced one mercaptohexanol. It works as a blocking agent. We optimized the blocking buffer finally to get the sensing signal. It turns out to be 10 millimolar of PPS, of pH 7.5, 5 millimolar of MgCl2, and 0.1 millimolar of EDTA. This is just supporting data. So we did aptamer in aflatoxin B1, and finally aptamer in presence, aptamer in aptamer folding buffer and in presence of arsenite. Okay, so again, so we got two minimum, 245 millimeter and 260 millimeter. That kind of, by this group, they shows that is parallel and anti-parallel. Anyway, it is the supporting data. We need finally a sensing signal. Okay, so this negatively capped gold nanoparticle, it is cited capped gold nanoparticle, was synthesized using modified Turkevich method. This is the synthesized gold nanoparticle. This is a transmission electron microscope image. The scale bar is 100 nanometer. This is the UVV spectra, and the peak is around 525 nanometer. And the size distribution shows it has a average value of 12.4 nanometer. Now we need to bind this gold nanoparticle on the optical fiber. Fiber was silica, so it has silanol group after treating with H2S4. So we treat the fiber with aminosilane, and finally it expresses this amine group on the surface. Now the rest is just we need to dip the fiber in the gold nanoparticles, and when we dip the gold nanoparticle fiber on the gold nanoparticle, we get absorbance like this. It is a real-time binding, and this okay. So I have two graphs. One is this is x-axis is wavelength and y is absorbance. At the nanoparticle binds on the surface, we get absorbance increase, and there is a peak at the same place. And this is a time-dependent graph. So after 300 seconds, we wash and we see that the surface is bound by gold nanoparticle. So this is a FXM image of gold nanoparticle coated surface. And the the scale bar is 100 nanometer. Okay, similarly, we also synthesized cysteamine cap gold nanoparticle. So it was synthesized by reduction of using NaBH4 in presence of cysteamine, and we use gold salt for the synthesis. This is how it looks, and it has a absorbance maxima in UV visible around 240 nanometer. This is a TAM image, and this scale bar is 50 nanometer, and it has a average diameter of 25 nanometer. And this is elemental composition by FECTEM. It shows presence of nitrogen and carbon, which indicates that the nanoparticle is coated by cysteamine. It is the nitrogen indicates it is coated by cysteamine. Okay, now the binding of cysteamine cap gold nanoparticle on silica nanofiber is quite difficult. So literature reported to treat the fiber using CDI, carbodiimidazole, but we saw that even after doing that, it was not coating properly, uniformly. So we further with what we thought, why not we use the same amine group on both ends. So basically we treated the optical fiber using siliconization again. So now we have amine group on the surface, then we use CDI, and finally we bound the system in cap gold nanoparticle, and this is kind of covalent interaction we are thinking that happening. Okay, so this is the binding. So after binding, we get such kind of peak. After rushing, it is not coming out, and this is the FXM image that shows quite uniform binding on the surface. This is 100 nanometer of scale bar. Okay, so coming to the sensing result. So the aptamer has thiolanded. So the optical, both of the cited cap and system in cap gold nanoparticle coated fiber was incubated in aptamer for 6 hours. And finally, it is supposed that such kind of bonding is forming, that it was reported by this Burgi group in nanoscale 2015. What we observed that, okay, now, okay, first I am explaining the graph. So it has a x-axis, which is time, and y-axis is absorbance. This control means we are dipping the fiber, ready to use fiber in the solution and checking whether it is stable or not. So this means stability. And the error bar is basically from 3 different probes used in 3 different days. And the standard deviation is giving the error bar. When we dip this probe having cited cap gold nanoparticle, we didn't find any increase of absorbance. It rather goes down like this. However, for system in cap gold nanoparticle, we got for 1 ppm, we got such response. And so we explored more and we found that for 1 ppb, 10 ppb, and 100 ppb, we are getting increase of absorbance. The linear plot is plotted like this. The x-axis is actually log scale. So, and we get a R-square of 0.97. And we can clearly see the control error bar. Okay, the first concentration is at least 3 times higher than the control error bar. So we can say that we can go up to 1 parts per billion of sensing. Okay, so the aptamer binding on the system in cap gold nanoparticle was also verified, let's say, by secondary method. So first, it was tested with hydrodynamic diameter testing, so using DLS. So the system in cap gold nanoparticle has a hydrodynamic diameter of 31.92 nanometer. After it is treated with the aptamer, obviously the aptamer is going coating on the surface, and it increases the hydrodynamic diameter to 55.7973 nanometer. And also zeta potential of system in GNP, as it is positive. So we found it was around 34.2 millivolt. After aptamer treatment, we are getting negative. Obviously the aptamer backbone is negative. That's why we are getting the negative result, negative transfer of zeta potential. Both increase basically indicates indirectly that the GNP has been coated with aptamer. Okay, so then the sensing was tried in real matrices. We have used tap water and lake water as the real matrix. Okay, so for tap water, this is the control, and we have spiked both of the water to check whether it is giving a similar response or not. First, the control was found to be stable in both the water. Then we have again spiked with dip the sensor in 1 ppb, 10 ppb, 100 and 1000 ppb of water. This is the linear plot of R-square of 0.97. For the lake water, we found similar response with the R-square of 0.98. In sensor, there is something called matrix factor. That is defined by slope of the calibration curve in presence of matrix divided by slope of calibration curve in absence of matrix. When it is in between 0.85 to 1.15, then all it is claimed that your sensor is basically considered working in the matrix. Otherwise, it could be because the matrix itself. So we calculated the same for the tap water. We found it was around 1.815. For lake water, it was kind of 1.009. Interference study was thoroughly studied with 1 ppm of mercury, copper, sodium, phosphate, chloride, sulfate, carbonate, Ciprofloxacin is an antibiotic, and enrofloxacin also fluoroquinolone antibiotic. And this is the bar diagram for arsenite. And this is the bar diagram for all of the interference added together. So it kind of stays in the same range. And this is the comparison graph. So this is the change of absorbance in DI due to 1, 10, 100, and 1000 ppb. In tap water and lake water, the response is slightly higher. That could be due to whatever these interferences are adding up in the sensing. Okay, so that brings to the last slide. So in conclusion, we have been able to optimize a LSPR-based event optical fiber sensor, which works on evanescent wave-based sensing method that can detect arsenite as low as 1 parts per billion, which is below the allowable limit by WHO, which is 10 parts per billion. And it has a linear range of operation from 1 ppb to 1000 ppb. The operation time is 30 minutes. Cost of detection is just 25 cents. Okay, but the arsenite main contamination is in the groundwater. But groundwater is very complex matrix, and also it varies from location to location. Like groundwater has a interference of 300 ppm of carbonate and 100 ppm of sulfate. So we are slowly moving to make a groundwater mimic to see that whether we are getting similar response or might be enhanced response due to the interference, so that we can finally go to the groundwater real sampling. So yeah, so I would like to acknowledge my Department of Science and Technology India to provide me INSPIRE fellowship for my PhD, and IIT Bombay for giving the research facilities. And that's the video I was talking about. So that's the probe. We put it in the holder like this, and it is basically 3D printed in our lab. And there's a click, so it gives a hydrophobic region. Sorry, I mean waterproof, leak-proof region. Then the cartridge is inserted in the device like this. The light you saw was basically amber light. Then we drop the sample over here. And as it is optical fiber paste, we then close the lid to give it a dark region. And finally, we set the zero value and wait for 30 minutes for the absorbance to go up. And finally, we take the reading. Thank you, guys.